Ever thought why we can't remember our earliest memories? Today we dive into the fascinating world of infantile amnesia. Just think about it. You, me, virtually every human on Earth, we all share a common blank spot in our memories. It's as if the stage curtain of our life story lifts only around the age of 2 or 3 years, leaving the earlier acts shrouded in mystery. This universal memory blackout is known as infantile amnesia, a widespread yet often overlooked form of memory loss. It's not that we didn't form memories during our infancy. Rather, it's that we can't access them. Imagine your earliest memories. Maybe it's a birthday party or your first day at school. Now try to recall something from when you were a baby. Chances are, you can't. You might think it's because nothing particularly memorable happened. But consider this: learning to walk, uttering your first word. Aren't these milestones significant enough to be etched in our memory? And yet they're not. So what's going on? Why do our brains lock away the memories of our infancy? It's a question that has intrigued neuroscientists and psychologists for decades. Some have suggested that biochemical processes could be at play, but a recent study from Trinity College Dublin offers a fresh perspective. It explores the role of a mother's immune system in this enigma. The researchers hypothesized that maternal immune activation, or MIA, which has been linked to conditions like autism spectrum disorder, might influence infantile amnesia. Their experiments involved conditioning young and adult mice to fear an electrical shock, and they made some intriguing discoveries, especially related to male offspring born to mothers with immune responses during pregnancy. These findings, along with further research, could shed light on the complex interplay between a mother's immune system and a child's memory development, possibly offering a potential link between early life experiences and conditions like autism spectrum disorder. So, what causes this unusual phenomenon? Let's unravel the mystery together. Researchers have long hypothesized that biochemical processes might play a significant role in infantile amnesia. The human brain is a complex organ, a labyrinth of interconnected neurons and synapses, constantly firing and rewiring. Within this intricate network, biochemical processes are ceaselessly at work, shaping our thoughts, emotions, and memories. The consensus among the scientific community is that these biochemical processes do indeed contribute to infantile amnesia, the phenomenon where we are unable to recall our earliest memories. Memories are not simply stored and retrieved like files in a computer. They are dynamic, constantly changing, influenced by a myriad of factors, including our body's biochemistry. Imagine a vast sea of neurons, with memories as islands scattered throughout. The biochemical processes are like the currents, the tides, the winds, shaping the contours of these islands, determining which remain visible and which are eroded away, submerged beneath the waves of forgetfulness. But what if there's another player in this complex game of memory formation and retention? What if our mother's immune systems also have a role in this process? It's a fascinating concept, isn't it? The idea that our ability to remember or forget could be shaped even before we're born, in the womb, influenced by the biochemical milieu that our mothers provide. This study from Trinity College Dublin proposes just that. It suggests that maternal immune activation, or MIA, could impact infantile amnesia. MIA has been associated with conditions like autism spectrum disorder, and now it might be linked to our earliest memories too. Here's the crux of it. The researchers hypothesized that a mother's immune system, specifically a small immune protein called cytokine IL-17A, might influence the formation and accessibility of memories during infancy. It's a bold assertion, one that could transform our understanding of memory and development. Could our mother's immune systems be the key to unlocking our earliest memories? This study from Trinity College Dublin suggests so. It opens up a new realm of possibilities, a new avenue to explore in our quest to understand the enigmatic phenomenon of infantile amnesia. The researchers at Trinity College Dublin decided to test this hypothesis using mice. Their experiment was designed to investigate the potential link between maternal immune activation, or MIA, and infantile amnesia. The participants in this study were not humans, but mice, both young and adult. The experiment involved conditioning these mice to associate a particular environment with an unpleasant experience, an electrical shock. The mice were then reintroduced to the environment without the shock. The researchers monitored their reactions, which would be indicative of their memory of the unpleasant experience. Here's where things get intriguing. The offspring of the mice whose mothers had immune responses during pregnancy showed a different pattern of memory retention. These mice were male, and they displayed signs of social behavior deficits that are typically associated with autism spectrum disorder in humans. But what's truly fascinating is their memory retention. These mice exhibited a prolonged retention of fearful memories. In other words, they remembered the fear associated with the electrical shock for a longer period than the control mice and their female siblings. This suggests that the maternal immune activation during pregnancy had somehow influenced their memory formation, making their early life memories more accessible. To probe deeper, the researchers employed transgenic mice with labeled memory neurons. This allowed them to visualize the engrams or memory traces in the brain. They discovered differences in the size and structure of these engrams in the dentate gyrus. This area, located in the hippocampus, is critical for memory formation. The differences were seen in the mice born to mothers with immune responses during pregnancy, suggesting a potential role of the mother's immune system in shaping the child's memory. These findings suggest that a mother's immune response during pregnancy might affect her child's memory formation. It's a fascinating look into how our earliest memories may be shaped, and why some of us may not be able to access them. It's an important piece of the puzzle in understanding the enigma of infantile amnesia. But what's behind these effects? The researchers believe a tiny immune protein called cytokine IL-17A is a key player. This protein, produced by the immune system, appears to have a significant role in memory formation and retention, specifically in the context of infantile amnesia. The researchers made this discovery while examining the brains of transgenic mice. These mice were genetically engineered so that their memory neurons, or engrams, could be visibly labeled. This allowed the scientists to observe and analyze the size and structure of these engrams in a region of the brain called the dentate gyrus. This region is nestled in the hippocampus, a part of the brain we know to be critical for memory formation. What they found was fascinating. There were clear differences in the size and structure of the engrams between the mice born to mothers with active immune responses during pregnancy and those without. The engrams in the mice born to mothers with an activated immune system were larger and more complex in their structure. But here's the kicker. The researchers then looked at mice born to mothers who lacked the protein IL-17A but were subjected to the same immune responses during pregnancy. Despite these immune responses, the mice still experienced infantile amnesia. This suggests that the presence of IL-17A in the mother's system during pregnancy might be critical to the formation and accessibility of early memories. So what does this mean? It appears that the protein IL-17A, produced by the mother's immune system during pregnancy, might influence how the offspring's brain forms and stores memories. This is a groundbreaking discovery, as it suggests the mother's immune system could potentially play a significant role in the development of conditions like ASD and in the phenomenon of infantile amnesia. This tiny protein, Illinois 17A, might seem insignificant in the grand scheme of things. But imagine if it holds the key to unlocking the mysteries of our earliest memories. So it seems that this small protein might hold the key to our understanding of infantile amnesia. Understanding the mechanisms behind infantile amnesia isn't just a matter of satisfying our curiosity, it has important implications in education and medicine. As we navigate the uncharted waters of infantile amnesia, it's not just about unlocking the secrets of our earliest memories. It's about understanding how these mechanisms could impact key areas of our lives. Imagine the possibilities in education. If we can understand why and how we forget our earliest memories, we may be able to enhance the way we teach and learn. By understanding the intricacies of memory formation and retention, educators could potentially tailor teaching methods to make learning more effective and long-lasting. In the realm of medicine, the implications are no less significant. This field is no stranger to the mysteries of the mind, with conditions like Alzheimer's and dementia taking center stage. But what about disorders that originate much earlier, like autism spectrum disorder? Could understanding infantile amnesia provide us with insights into these conditions? The Trinity College Dublin study suggests a potential link between early life experiences, maternal immune responses, and the development of conditions like ASD. It's a tantalizing glimpse into how our earliest moments may shape us in ways we're only beginning to understand. If these connections hold true, we may be looking at a paradigm shift in how we approach and treat ASD. The understanding of how a mother's immune system can influence a child's memory development could lead to early interventions that could potentially mitigate the effects of the disorder. But it's not just about ASD. The findings of this study could have far-reaching implications for other conditions linked to memory and brain development. The study offers a glimpse into the complex interplay between a mother's immune system and a child's memory development, shedding light on why some memories are accessible while others remain elusive. It's a fascinating journey, and we're just at the beginning. Who knows what other secrets we'll uncover as we delve deeper into the mysteries of our minds?